Okay, so this is what you will need to do for the Catalase lab, the enzyme Catalase lab. So I'm making the video right now. So this is the document which talks about what you're going to do. A lot of the things are similar. Don't need a cover page, page number, headings in bold, binomial name. Okay, you're still using potatoes, so hopefully this helps. However, because this time I'm more looking at your design component as well as your result data processing. So in this case, there will be some new stuff, there will be some old stuff. So let's take a look. In terms of, you will need to have a research question. Okay, you can use the template that is provided. You will need to have a background. Now, you guys had done a background already. Um, if you look back on that day where I wasn't in and you guys were, yeah, when I wasn't in and you guys were working in groups, you guys were kind of doing a research on what is going to be on the lab about catalase and whatnot. So use that. Now, however, you can use that, but you can't just directly copy and paste it in, because at that time you didn't know what was your independent variable, you didn't know what your dependent variable, and you kind of didn't know how the lab was going to happen. So in this case, you will need to add that in. You will need to explain a little bit more, talk a little bit about your independent variable. Why is it important? Talk about your dependent variable. Why is it important? Talk a little bit about your context. So why is catalase in potato, for example? And in this case, how would the lab basically work? How would measuring the foam would be a useful way to show measuring your dependent variable. So talk about that. Okay. Since you have done research for it, you will need to do in-text citation. Now, one thing I do notice, in-text citation, you have to use in-text citation whenever a new idea is talked about. So as long as it's something that we haven't talked about in class, but it's something that you looked up to talk about, make sure you have in-text citation. It's not only for direct quotes, okay? Even paraphrasing require in-text citation. So make sure you do that, and make sure you only use reliable academic sources. Britannica works well as a number of other websites. Please don't use anything like Khan's Academy or some kind of tutoring kind of website, okay? Academic sources. Now, in terms of these other four parts, you will need to do that. Now, in terms of what you need to have in there, I would ask you to click on this link. This is a lab format guide. I've written it a while back. And as you can see here, it has a lot of details in terms of what you need to write. For example, what do you need to write for your independent variable? What do you need to write for your dependent variable? How are you going to write your controlled variable? We kind of talked about that in the last few lessons when we're planning this lab. Now, this part here, uncontrolled variable. Now, I have a lot of questions about that. What is an uncontrolled variable? So even though you've done an excellent job in terms of controlling variables such as the concentration of the hydrogen peroxide, for example, but there are stuff that you weren't able to control. So for example, and I'll give you this one, and you can't use it. Okay, you can't use that as a you can't use that in your or that doesn't count. So in this case, the room temperature. The room temperature, you don't have any control on it, but how would that affect your result? It would affect your result because temperature is a component that affects the.
Will the Q come to the main office?
Yeah. Temperature is a component that will affect rate of reaction and the fact that your hydrogen peroxide has been in room temperature, that would certainly affect the result. How are you going to monitor the change? Well, we have a thermometer, and we did a measurement of the room temperature, and Mr. Cheng put it on the board that morning. Okay, so make sure you look back at that. I did tell people to record that number. So this is what you need to do for uncontrolled variable. Now, a very common question: how many controlled variables do I need? I would say for this one, you should list as many as you can think of. You need to list. There are no minimum number. You need to think about what there is and list them out. Okay. Apparatus, method, and procedure. Make sure you follow the instruction over here, what to write and how to write it, for example. In terms of the safety and ethics, now I don't need you guys to do environmental concern for this one, just for the simplicity of it. But right now, you will talk about you will talk about safety as well as ethics. Okay. So, are you, for example, working on some rare endangered species? If you are, then that would be an ethical concern, and how would you minimize that risk? If you are not, then say it. There is no ethical concern using the species that we're doing. The idea is that getting you guys to be used to writing out safety and ethical concern. This is very important, especially if you're doing any experiment with live animals, including humans. That is important. Okay. So don't worry about the environmental concern, but do have safety and ethics. In terms of data processing, a lot of it will be similar. The one big thing that will be different is the following. You will still need collected data. You will still need qualitative observation. You will still need process data table and sample calculation. However, in this situation, your processing, what you should have, you're only using your own group. You're only using your own group's information. So you should have the averages of each. Let me try to remember. You guys were doing each variation of IV. Okay, so if you were doing temperature for those 4 different time, how long you dip your potato in the hot water bath, you have the average of those 4 trials. Now, this time, I also want you to do standard deviation. I also want you to use Excel to calculate standard deviation. How do you do that? Well, there are a lot of videos how to do standard deviation on Excel. Pretty sure they're listening in on this. So there are a lot of videos in terms of how to do that. I will suggest you look up online to do it. Okay. So find the one that works for you, look it up. In terms of graphs, what I will want you to have is you should have the trend line. Now this time you should have one line, trend line. You should have R2 value, and you will need to use error bar. And your error bar should be standard deviation. Okay, should be the standard deviation of that variation. What I mean is this. So I'm going to use. Now I have also a sample paper here. I haven't looked at this sample paper in terms of the data processing, so everything else looked really good. Yeah, so you see they have They were doing something with pHs. They have averages. They have standard deviation. Don't worry about this correlation coefficient. Don't worry about that part. But as you can see here in their graph, for example, because look back at the table. Now, because if you look at pH of 4, the average is this, but the standard deviation is huge because the data is really widespread between those 5 trials. But for 4.5 pH, the data, all 5 trials, their data weren't really that spread out. So the standard deviation is small. So what it means is that you will need to input individual error bar for those points. Okay, you will need to do that. Now, how to do that? There is on this format guide here, there is a method in terms of how do you, this video here will tell you how to calculate the standard deviation and add them onto the error bar in Excel. So make sure you watch that video and and follow it. Okay. So the sample paper and the format guide, you will need to use a format guide quite a bit. Make sure you use published sources. And this is the same as before. Make sure you have citation. Now, I also noticed in the osmosis lab, some people actually put things in works cited, but it wasn't mentioned or it wasn't cited in the writing. Okay. In APA writing, you only put things that you have actually in text put in text citation. Otherwise, if you read it, but it is for your inspiration only, you do need to still talk about it, okay, in your background. So the sample paper, I do want to talk about a little bit about this. The sample paper, this is a full lab write-up, and this is also a lot of pages. This person had an extensive background, a page and a half, 1.5 fonts. You don't need to have that much, okay? I think probably half a page to a 3/4 of a page would do. Half a page for you guys, half a page and 3/4 of a page would do. But I do expect to see in-text citation there. I'm not asking you guys to do hypotheses simply because once you do hypothesis, you should technically do alternative hypotheses. So I don't want to get on that part. But as you can see here, independent variable, there is some variation in terms, they didn't say what unit it is, but the rest are pretty similar to what I asked. And their control variable table is also very similar. They just moved the control methods here and justification here. As you can see, they talk a lot, quite a bit. And uncontrolled variables, they had 2. Now keep in mind, it's a different experiment. So the material lists, as you can see, they do show the uncertainty for each instrument. So if you kind of don't remember certain things, on Monday or Tuesday, come back and just ask me to show you the instrument itself. The methods, step-by-step written, is very clear. They have a safety, ethics, and environmental impact. Don't worry about the environmental part, as I said. Qualitative data. Now, in terms of qualitative data, you see that they only put in pictures when it's actually useful. And also, they will cite the pictures in terms of they'll write a little blurb about the picture and what's so important about it, so that it seems to me it's not a page filler, so to speak. Your raw data tables, process data, sample calculation. There's a lot of calculation you don't need to do for this one. and then a graph. Okay, they have the units on there, as well as the equations. I don't need you guys to write these. You don't need to write the standard deviation part. I would probably want you to write 1 or 2 lines just about why did you pick that type of trend line. Yeah. So that is it in terms of, oh, they also have an appendix. You don't need to do that. And this is the works cited section. So as I said before, this is a good sample lab write-up. So you can use that to take a look, kind of the detailed and how does it look like. Let me know if you have any questions. Now, the deadline. The deadline will be on Wednesday. It will be on Wednesday, November 9th. Okay. As you can see, the e-copy is earlier that morning, 6 a.m. Okay. You will need to hand in the hard copy in class, in person in class. Okay. I just have the electronic copy submission, so I don't have people coming in and say, sir, I just still need to upload it. You should have uploaded it before you show up to class. That's why I put it there like this. Okay. So deadline, November 9th, Wednesday morning in class, hard copy in class, e-copy 6 a.m. If you want me to look over your draft, you will need to share your doc with me by Monday morning around 8 or 9, because this week is a week where all the teachers will be very, very busy. I do need you to share that with me early so that I have enough time to read and to get back to you. Okay? So hopefully this helps. If you have any other question, please let me know. Otherwise, good luck.